Hello students. In today's session, we will discuss a very interesting concept, that is packaging of DNA. Before we move on to the concept, we will discuss some few interesting facts, such as the length of human DNA, one, the size of human cell, and the size of nucleus. And you already know that the DNA in case of eukaryotes is confined to the nucleus. Confined to the nucleus. The length of human DNA, diploid content of human DNA, is about 2.2 meters approximately. It is about 2.2 meters. And the size of the human cell or typical cell is about 100 micrometers, and nucleus is a minute structure even smaller than the cell. It is about 6 micrometers. The next question arises: how such a long polymer accommodate within the cell, that too within the nucleus? For example, in case of diploid content of human cell, there are 46 chromosomes. Within the chromosomes, DNA is coiled or genetic material is present. If you take out the DNA from all the 46 chromosome, the length of diploid content of human DNA is about 2.2 meters. The next question arises: how the packaging of DNA takes place in case of prokaryotes and eukaryotes? Before we move on to the packaging of DNA, first we'll know how we can calculate the length of DNA. Let us find the answer. So, the length of DNA, how we can calculate the length of DNA? Is simple. The length of DNA calculated by multiplying the total number of base pairs with a distance between two consecutive base pairs. And you already studied the Watson and Crick model of DNA. In case of bDNA or DNA which is coiled in a right-handed fashion, the one complete turn is made up of 10 base pair, and the length of one complete turn is about 3.4 nanometer. And one complete turn is made up of 10 base pairs, and the distance between two successive base pair is about 0.34 nanometer. 0.34 nanometer. So the distance between two consecutive base pairs in case of the DNA which is coiled in a right-handed fashion is about 0.34 nanometer. And the total number of base pairs present in case of diploid content of human DNA is about 6.6 into 10 raised to 9 base pairs. Let us find the length of the human DNA. So, you already know the formula. Total number of base pairs is multiplied with the distance between two base pairs. Let us, and one more thing you remember, okay, it's very easy to calculate, convert a nanometer into meter. Okay, if it is multiplied with 10 raised to minus 9, so nanometer is converted into meter, 1 billionth of nanometer. Okay, nanometer is 1 billionth of meters. So the total number of base pairs in case of diploid content of human DNA is about 6.6 10 raised to 9 is multiplied with the distance between two consecutive base pair is 0.34 into 10 raised to minus 9. So we'll get the length of diploid content of human DNA is okay, that is 2.2 meters. Okay. In case of haploid content of human DNA, the length of DNA is 1.1 meters. So because in case of haploid cell only 23 chromosomes are present, whereas in case of diploid cell, diploid human cell, there are 46 chromosomes are present. So diploid content of the length of diploid content of human DNA is 2.2 meters, whereas the length of haploid content of human DNA is 1.1 meters. And we'll look at the other example. In case of prokaryotes, typical prokaryotes, that is E. coli, the length of DNA in E. coli is approximately 1.36 meters. The next question arises: how is such a long polymer accommodate within the cell or packaged within the cell? Either it may be in the prokaryotes or eukaryotes. So next we'll move on to the packaging of DNA in case of prokaryotes. So you already know the difference between prokaryotes and eukaryotes. In case of prokaryotes, well-defined nucleus is absent, whereas in case of eukaryotes, nucleus is present. Well-defined nucleus is present. That means in case of prokaryotes, the DNA is not scattered throughout the cell. Okay. Even though nucleus is absent, it is not scattered throughout the cell. The DNA being negatively charged and is organized in large loops with some positively charged protein in a region called nucleoid. So in case of prokaryotes, okay, DNA is organized into large loops with some positively charged proteins in a region called nucleoid. So this is a typical cell where the nucleoid is present. Within the nucleoid, DNA is present. Next we'll move on to the DNA packaging in case of eukaryotes. You remember, in case of eukaryotes, packaging of DNA carried out with the help of specialized or positively charged basic proteins called histones. Okay. So you already know that DNA is associated with proteins in case of eukaryotes, and the packaging of DNA is carried out with the help of positively charged basic proteins called histones. And you already studied in first year, there are 4 types of amino acids, or there are 3 types of proteins: acidic proteins, basic proteins, and neutral proteins. Okay, neutral proteins. Acidic proteins are made up of more number of acidic amino acid. Basic proteins are made up of basic amino acids, whereas neutral, equal number of acidic amino acids and basic amino acids. So I'll not discuss in detail about those amino acids. In case of eukaryotes, it takes place with the help of basic proteins, and those basic proteins are called histones. And the basic proteins are positively charged. Okay. Basic proteins are positively charged, and these basic proteins, the basic proteins are rich in 2 amino acids. One is lysine, another one is arginine. Lysine and arginine. The histone proteins are rich in basic amino acids. The 2 basic amino acids are rich in histone protein. One is lysine, another one is arginine. Already I told you. Look at the picture. The red color one are histone proteins, whereas blue color one are the DNA. That means DNA is negatively charged, whereas histone proteins are positively charged. That negatively charged DNA wrapped around the positively charged histone proteins during packaging. Let us start with a histone protein. We will discuss about histone proteins. The 5 classes of histone proteins are associated with the packaging of DNA in case of eukaryotes. H2A, H2B, H3, H4, and the last one is H1. So 5 classes of proteins. Out of these 5 classes of proteins, the 4 classes of histone proteins occur in pair. That is, H2A occur in pair, H2B occur in pair, H3 and H4 also occur in pair, and together organized to form histone octamer, because there are 8 proteins, histone proteins are involved in packaging of DNA, and this organized structure we called as a histone octamer. So this is one histone octamer, and this is second histone octamer around which DNA is wrapped. And look at here, in between the two histone octamer you find the DNA, and that DNA we called as a linker DNA, or we called as a spacer DNA. Let us start. So this is and in between the two histone octamer, one more protein which is involved, okay, that is histone protein. So there are 5 histone proteins are associated with the histones molecules are associated with the packaging of DNA: H2A, H2B, H3, H4 and H1. So in human cell, 2.2 meters of DNA in the 46 chromosomes. How the first order packaging takes place? Let us discuss. Negatively charged DNA wrapped around the positively charged histones. This is histone proteins around which negatively charged DNA, and the diameter of this histone octamer is about 2 nanometer, and this part of DNA composed of 146 base pair. That means, so the DNA which wrapped around the histone octamer is made up of around 146 base pair. Okay, I'll repeat once again. The DNA which is wrapped around the histone octamer is consist of 146 base pair. 146 base pair. Okay. The form, and this we called as a new bodies or nucleosomes. And you remember, okay, nucleosomes are the basic structural unit of chromatin fiber. So each nucleosome composed of 2 components. One is DNA and second one is protein, histone protein. Then the other histone protein, okay, called H1, which lies outside the nucleosome, okay, outside the bead or outside the nucleosome, and this protein binds to the DNA of the nucleosome where it enters and lives. So here exactly in this position, I'll show you. In this position, H1 histone protein is present, through which DNA enters and leaves the nucleosome or new bodies. So what is the function? The significance of H1 protein is, histone protein is, it stabilizes nucleosome structure and also helps in proper packaging of nucleosome into polynucleosomes. So when many nucleosomes join together to form polynucleosomes, and this H1 histone protein which helps in packaging of nucleosomes. Okay, one, the function is it stabilizes. Second one, it helps in packaging. So look at here, the DNA connecting two adjacent nucleosome. So in between two nucleosome there is a connecting DNA. That connecting DNA we called as a spacer DNA or linker DNA. Linker DNA. So its length is variable and it extends from one nucleosome to the another nucleosome. This linker DNA is made up of about consist of 60 base pairs. 60 base pair. When you look at the structure of typical nucleosome, the typical nucleosome is made up of 200 base pairs. Approximately it is made up of 200 base pairs. So nucleosome constitutes the repeating units in a thread-like structure called chromatin. So when many nucleosomes arranged like a beads on a string, and which is called as a chromatin, and this bead on a string is visible only under electron microscope. And the nucleosome repeated every 200 base pairs, that is 140 which 140 base pair which coils around the histone octamer or nucleosome, and the remaining 60 base pairs which linker DNA. Linker DNAs, the base pairs are present in the linker DNA. And the chromatin is condensed to form chromatin fiber or packed to form chromatin fiber. Look at here, this is the first level of packaging. It condensed to form chromatin fiber that are further coiled and condensed metaphase stage of cell division to form chromosomes. And you remember one thing, the condensation or the coiling of packaging of DNA at higher level requires additional set of proteins, and those proteins are called non-histone proteins. Normally, there are five class of proteins associated with the packaging of DNA. Along with these five proteins, one more protein that is called non-histone protein, which helps in the condensation of or coiling of DNA within the chromosomes. Then the typical eukaryotic chromosome of about 10,000 base pairs of DNA will have 50 nucleosomes. So very simple to calculate the total number of nucleosome. If we divide the total number of base pairs with the number of base pair present in each nucleosome, that is 200 base pairs, will get the total number of nucleosome present. The total number if you calculate the total number of or divides total number of base pairs divided by the number of base pairs present in nucleosome. So total number of base pairs present in typical nucleosome is about 200 base pairs. So we'll get the total number of nucleosome present in the given DNA. So this completes the packaging of DNA. So in some region of nucleus, the DNA is tightly packed. In some region it is loosely packed. So based on this, there are two types of chromatin fibers are identified. One is heterochromatin, another one is euchromatin. Look at the diagram. There are two stains are there. One is lightly stained, white color one. Other one is darkly stained. The one which darkly stained is called heterochromatin. The lightly stained one is called euchromatin. Then, so this is the heterochromatin. Okay, then this is loosely packed one is called euchromatin. The other difference, heterochromatin is transcriptionally inactive, whereas euchromatin is transcriptionally active. So transcription means what? The process of synthesis of mRNA from DNA is called transcription. That means the RNA is synthesized from the DNA. RNA is synthesized from the DNA. There are three types of RNA: mRNA, rRNA, and tRNA. And all these RNA are synthesized from DNA by the process called transcription. So when you compare chromatin fiber, heterochromatin is transcriptionally inactive because it will not synthesize any RNA, whereas euchromatin is transcriptionally active. And the last difference is the heterochromatin replicates late, whereas euchromatin replicates early. So this completes the packaging of DNA. So in the next session, we'll discuss the search for genetic material. Okay. Thank you.